areas that I'd like to look at with you is a topic that you're going to be studying at whatever level that you'll be with us at, whether that's level 1, 2, or 3, in the subject of cell biology. So we'll look at what cell biology is, and we'll also delve into as well the idea of cell theory. We'll build on that, and we'll move over to the third point in our session, which is looking at the measurements that are used in cell biology. And then we'll start to focus a little bit more in section 4 as the two main groups of cells. We can see those a bit more in sections 5 and 6, and we'll start to explore the topics in a little bit more detail of prokaryotic cells, and in topic 6, eukaryotic cells. We'll have a little bit of a quiz, just as a summary, and then I'll provide you with some links for some further research and reading of the topic. And then finally, in section 9, what we'll do is I'll give some guidance about your plans for 2021, the options that are available to you, and if you're stuck or you need some more guidance or information, some contact details. So I hope you find it of interest and of use. So our first area that we're going to look at is what really is cell biology. Well, cell biology comes from the point of considering that the cell is the most fundamental unit that's available, and as a life form. It's not a new science. Really, Hook proposals back in 1665 looking at plant cells, followed on by Anton van Leeuwenhoek's work in 1677. Some of the most primitive microscopes started to identify what we now understand to be a cell. Of course, these days we don't look at cells with primitive microscopes. I'm sure in college and schools you've been used to using light microscopes, which are very effective and can easily look at magnifications of 1,000 times. And in the diagram, in the photograph, that you can see that's an electron microscope, and that can go to millions of times magnification, and so that we can see the superstructure, the real detail within the cell, and what makes a cell. We ask the questions: if all cells are the same, if they're different, and if they're different, why, and what are their components. So with that in mind, then we come to a topic that you're familiar with during your course, and we refer to as cell theory. So what is this thing called cell theory? So cell theory really has three main concepts, and it tries to suggest to us that it's a universally accepted theory, but it tries to suggest to us that all living cells, all living organisms rather, are made up of components referred to as cells, and these are the basic organisational unit of life. And the cells originate from pre-existing cells that divide and that replicate. So what's the history of it? Because it is quite unusual.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. 1. All organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day, something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
Okay, so now that we're trying to move on and start to consider a feature that will become very prevalent in all of your studies, and it's the units of measurements. Of course, we're familiar with units of measurements used in most areas of life, whether it be centimeters, meters, kilometers and things. When we start coming into cell biology, we have to go smaller. So let's have a look on the right hand side, and what you can see there is our larger units of measurement, the meter and the centimeter and the millimeter. And then as we move down, we come on to perhaps two units that you won't be familiar with, the micrometer and the nanometer. And if we have a look on the diagram on the left hand side, sort of place where we see then the micrometer and the nanometer. Essentially what we start looking at is 1 micrometer is equal to 0.001 millimeter. It's clearly very, very small. And when we start looking into that area, we start looking at perhaps 100 micrometers, about the same size as human cells. And looking around that, but when we start then moving into looking at really small organelles, which is the chloroplast, and we can start looking then at using units of measurement called the nanometer, which is even smaller. And a nanometer can be used as a measurement of viral particles or proteins or even amino acids. If we want to get really small, then we can start looking at the size of atoms using a nanometer. So just in case you see those units of measurements, the micrometer and the nanometer, I'm sure you will, you've got some understanding of their size and proportion. So now let's have a look at the two main groups of cells that we come across. And these might be terms that you're familiar with, and if it is, that's great. If not to worry. First of all, we've got the cell group. It's a group of cells. These are cells that have certain common features. And the first one we refer to as prokaryotes. Now, prokaryotes have no nucleus, so they've got no membrane-bound, dense area of genetic material. They have no nucleus, and they also have no membrane-bound organelles. Now, the organelle is the complex subunit within that cell. And common examples, if you're ever asked about what an example of a prokaryote would be, it'd be a bacterium cell. And over on the right-hand side of the slide, in the top corner, you can see there you've got a cell of a bacteria. You can see that it's got a bacterial flagellum, which looks like a tail, and that's to help in locomotion, that's to help it move. And there's nothing, if you actually delve into the cell itself, as you can see, there's no complex membrane-bound organelles. Compare that to the diagram on the bottom, which then starts to look at a very complex eukaryotic cell. And that these are cells which have got a nucleus, and the nuclear material is contained within complex membranes. And you can see that they've got really complex organelles, really big names to them as well. Perhaps you've come across some of them before, perhaps you haven't, like a smooth endoplasmic reticulum, the rough endoplasmic reticulum, mitochondrium, centrioles, Golgi apparatus, cytoplasm, the cell membrane, which is there. So really complex groups. If you're asked to consider which type of cells would fit into the eukaryotic family, well, animal cells like our own would come into this group. They're complex, and they've got those membrane-bound organelles. We'd also look at plants, fungi, and protists as well. So it gives you a little bit of an idea. If you're going to be coming across those two terms during the year, what it refers to groups of cells. We can see there a pictorial form, a summary, really looking at the prokaryotes on the left. So an example of that would be the bacterium on the right-hand side. What we can see there is a more complex structure of the eukaryotic cell and everything that it brings. So let's have a little summary of that and just make sure that we've got a full understanding.
On Earth, there are two major types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the simplest and most ancient types of cells. They were the only form of life on Earth for billions of years before eventually giving rise to eukaryotic cells. If you take a look at prokaryotic and eukaryotic cells side by side, you can see a number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have a simpler structure. But bear in mind that this simple structure is actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you were to look inside a prokaryotic cell, you would probably be surprised at how simple it is. For example, prokaryotic cells lack a nucleus. In fact, the name prokaryote actually gives you a hint about its structure. Kary means kernel or nucleus, and pro means before. So basically the word prokaryote means before nucleus. The inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout its cytoplasm. The cell's DNA is located in a region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as a nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or a bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess one or more flagella, which are used for movement. In contrast, eukaryotic cells contain a membrane-bound nucleus and other membrane-bound organelles. The name eukaryote also gives a hint about its structure. Eu means true, and remember that kary means nucleus. So in essence, the word eukaryote means true nucleus. Eukaryotic cells are larger and much more complicated than prokaryotic cells. However, they do share a few things in common with prokaryotes. Let's take a look at a Venn diagram to compare prokaryotes to eukaryotes and to also see what areas they have in common. Let's start with their commonalities. Both prokaryotes and eukaryotes have DNA as their genetic material, have ribosomes, have cytoplasm, and have a plasma membrane. Now let's look at some of the ways they differ. Prokaryotic cells are the oldest type of cell. They're small and relatively simple. Eukaryotic cells evolved from prokaryotic cells later. They're larger and much more complex internally. Prokaryotes lack both a nucleus and membrane-bound organelles. Eukaryotic cells have a nucleus and also contain organelles. Prokaryotes are single-celled organisms. Eukaryotes can be either single-celled or multicellular. And finally, the DNA of a prokaryote is usually organized as a single circular chromosome, while the DNA of a eukaryote is organized as linear chromosomes.
There we go. So we can see then that prokaryotic and eukaryotic cells differ, but they also have some similarities. Now let's start looking at that level of detail. Let's start looking at that of a prokaryotic cell. As we said before, bacterial cells are probably the most common key example that people would give if they're asked to name a prokaryotic cell. They're very simple. They're single-celled organisms, so they have a degree of independence with how they function, and they lack that nucleus and that membrane-bound organelles. They're much smaller by not having the nucleus, because the nucleus takes up a large proportion of the cell. They're much smaller, and typical prokaryotic cells will range from 0.1 to 5 micrometers in diameter, and that's a lot smaller than the eukaryotic cells that normally go from about 10 to around about 100 micrometers. So it's worth bearing that point in mind. So much smaller. If we were to then go into the cytoplasm, so the fluid within that cell, and inspect it to see what it contains, one of the things that we'd see is that it doesn't contain any complex membrane-bound organelles, such as the mitochondria or a chloroplast or anything in that nature. And in fact, what we tend to see then generally things like ribosomes that we'll see in there. And there are two types of ribosomes that you'll come across in your studies this year, and then the ones that we'll see in a prokaryotic are the smaller ones that are referred to the 70S type. So sometimes people will say, well, bacteria doesn't have any DNA, it has no genetic material. Well, that's not true at all. It's most definite that a prokaryotic cell would certainly have genetic material. In the case of bacteria, DNA, but it'd be found really in a sort of a circular molecule within the nucleus, within the cytoplasm. It wouldn't be found within a nuclear membrane-bound system. And what we start to see as well is if we have a look at the cells, the cell walls, what we're going to find is often prokaryotic cells will have neuron as their main component, and this will help with structure. Prokaryotes as well will have something that's referred to as a plasmid. Now you may have come across that at some of the higher level of GCSE, and these are rings, circular structures of DNA that will contain genes, often associated to support the bacteria in antibiotic resistance. Now that's something that's quite important because it will provide then to this bacterial cell, these plasmids will provide a structure which will allow them for resistance against certain antibiotics. And that's not great for us as human beings, but certainly it's very helpful for the bacteria. So that we'll try and do things to prevent those plasmids being available. So we'll be cautious on how we use antibiotics. We'll make sure that we finish the course and we'll take them at regular intervals. But it's interesting then to start linking up that some of the science that we start to see is actually linked to our everyday life. So now let's move on to our sixth topic, where we look at the structure of eukaryotic cells and then the functions really of their components. So our eukaryotic cells are much larger, much more complicated than those that we saw from the prokaryotes, the bacterial cells that we discussed earlier. And we've got lots of these subcellular, much smaller inside structures, and we refer to those as organelles. And each organelle will have a specific function within the cell. And you'll see that not all eukaryotic cells are the same. And if they've got a special job, if they've got a special role, then the organelles within that cell will be very different. For example's sake, if you would be looking at a muscle cell, you'd be assuming then that there'd be much more mitochondria within a muscle cell, because you may know from your studies that mitochondria is the production of energy. So let's say on the diagram on the right-hand side, you can see a typical eukaryotic cell, very complex, with these membrane-bound organelles. So if I was to try and trick you then and try to suggest which diagram suggests as a eukaryotic cell, is it the one on the left, is it the one on the right? Of course, we'd be suggesting that both of the cells are eukaryotic. Just because they've got, they appear different is not an automatic indicator, so that they're not eukaryotic cells. If I was to ask you which one is an animal cell and which one is a plant cell, and knowing that animal and plant cells are both eukaryotic, I wonder if you'd know which one from there. Sometimes students mistakenly would have a look at the diagram on the right-hand side and see that it's got a mitochondria, and then suggest automatically that it must be an animal cell because only animal cells have mitochondria. Well, that's not correct. Both animal cells and plant cells will have the mitochondria, so we need to look in a little bit more depth than that. And instead, if we start to look at certain features on the on the diagram on the right hand side, such as like the cell wall, which gives structure. If we start having a look at the idea of chloroplasts, which is used in photosynthesis, if we start looking at the idea of starch grains and a vacuole, then we can start to really quite quickly suggest that the diagram on the right hand side is a plant cell and the one that's on the left hand side is an animal cell. But both are. So let's go in and have a look at some of the organelles found in eukaryotic cells. We'll have a little look at cell surface membranes, which you may have studied in your GCSE, but you probably only refer to those as just the cell membrane. You wouldn't give it the title of cell surface membrane. You just call it probably cell membrane. We'll have a look at the nucleus, the mitochondria, and then we'll dip into something specific with plants, such as the chloroplasts. We'll look at ribosomes. We'll also dip in as well to the cell wall. So when you were doing your GCSEs, you probably looked at a cell membrane, and it was something that you didn't give an awful lot of detail to and didn't give an awful lot of thought to. It is one of the parts of the GCSE syllabus that doesn't zone in. In fact, your cell surface membrane is a membrane that envelops around the cell. Particularly, we see them within animal cells. And what we're seeing is an incredibly complicated unit that's going to control what is actually going to come in and what is going to come out of the cell. It provides strength, it ensures that there's structure, and it acts as well as ensuring that there are receptors on the cell surface to then cause chemical reactions within the cell to make it react. So an incredibly important structure within any eukaryotic organism. We see the nucleus, and often, again in GCSE, the nucleus is just hand-drawn. It's got a dense area, often in the middle of the cell. It's a lot more complicated than that. It has a double membrane, so it's almost like a double wall going around the nuclear envelope. And then it has these little pores, these little channels, and they're referred to as nuclear pores. And what we then start to see is that the function of the nucleus, the real function, is then seen in a dense area in the center part of the nucleus called the nucleolus. And it's within the nucleolus really that we start to see where the genetic material, the dense genetic material, is contained. Mitochondria, as I'm sure that you've done in your studies in your GCSE, is looking at energy production, and you'll be looking at, certainly on your course, you'll start to look at a little bit more detail on what does energy production mean, and we'll start to consider the terms of something called aerobic respiration. We'll compare that to a similar term that you might have come across before called anaerobic respiration, and we'll see how that relates to the molecule called adenosine triphosphate, or ATP, and how that relates to a molecule called adenosine diphosphate, or ADP, and see what the relationship is between those molecules in order to produce energy. But it's an incredibly important organelle within a eukaryotic cell, and it helps then in that production of energy. And you'll spend quite a bit of time looking at that during the year. Of course, moving into plant cells, we start to look at chloroplasts, and that becomes really important, as the chloroplast is one of the main hubs. That's a site of photosynthesis within the cell, so the production of energy, but this time not from respiration, but clearly within a plant cell from light. So we'll see during the course about how that links. We'll also see as well with the chloroplast how the chlorophyll pigments, which are contained within the substructures of the chloroplast, about how they function to produce energy. Ribosomes are a very common organelle, often overlooked, very quickly. They're really made up of protein. They're very small, one of the smallest structures that we'll commonly refer to. Can't see them with a light microscope, not with a normal light microscope, but their main function is in the support of protein synthesis. You might remember from our slides when we were discussing prokaryotes that ribosomes within a prokaryote are common, and they're referred to as 70S, as in their size. However, with ribosomes within a eukaryotic cell are larger, and we refer to those as 80S. Regardless, though, they're all there for protein synthesis. We look at the cell wall, as I'm sure you know, your cell wall, which is very common in eukaryotic plant cells, but you can also find them in algae and fungi as well. Generally consist of cellulose mixed with various polysaccharides, those carbohydrates, and what they're doing, of course, is providing a structure, this waterproof structure that prevents cells bursting, because of course you've got the process of osmosis, that interchange of water molecules, so it's important then, particularly within a plant cell, that you've got this structure that's going to act to prevent that cell from bursting. So it's a bit of a whistle-stop tour then of the different components. I think the one thing that's important to bear in mind, especially if you're going to be studying this subject at colleges, we don't teach it very much in a way where it's going to be solely done by presentation or PowerPoint. You're going to have lots of practical work to do, lots of microscopy work where you can get hands-on. You'll make your own slides. You'll be making really good use of the microscopes to making sure that when you've finished, you've got great confidence with them. And if you're going to go onto the workplace, or particularly university, you're able to handle a light microscope without any problems at all. So let's have a little look at a quiz. See of some of the questions. So question 1, what are the 3 components of cell theory? Do you remember those from before? I'll give you a moment to have a think about that. Well, the 3 components, if you remember them, is that all living organisms are made up of cells. Cell theory also suggests as well that cells are basic organizational units of organisms, and that number 3, all cells come from pre-existing cells as they merge from that way. So let's see if we can remember them when we were talking about units of measurement. What's the smallest units of measurement? Is it a millimeter, a micrometer, or a nanometer? So have a think of those 3, which it is. Well, it's a nanometer. A nanometer is our smallest unit. So here we go for question 3. True or false? Prokaryotes do not have a nucleus, do not have a membrane-bound organelles, but do have a cell membrane and ribosomes. So what do you think? Is that true or false? Well, it's true. It's true. I'm hoping that you got that one correct from your studies from before. 4. Is a bacterial cell prokaryotic, or is it eukaryotic? What do you think? Prokaryotic or eukaryotic? Well, bacteria is prokaryotic because, of course, they do not have those membrane-bound organelles. What about plant cell? Are they prokaryotic or eukaryotic? Well, they're eukaryotic because, of course, they do have the membrane-bound organelles. And then finally, do you remember what we described the function of mitochondria? Well, we said it was the site of energy production by the process of aerobic respiration, where possible, and adenosine triphosphate, certainly within our animal cells. So further research and reading. If this is a subject that you've found quite interesting, or you'd like to do some more preparation, or even if you just want to study some more parts of science over the summer break, there's 3 good sites where you can go into. If you'd like to give some focus into biology, great site, some great reading all about biological systems and theories, so dig into that one. If, on the other hand, that you want to broaden your horizons a little bit more, maybe, because during the course you certainly won't just be studying biology, be looking at chemistry and physics. And if you want to start looking at some other courses or modules, FutureLearn actually has little built-in modules themselves that you can dip into. It's free, it's all free to access. So that's pretty cool. You can have a dip into that. And similarly, I'm sure you would have come across the Khan Academy as well. Some fantastic resources on there for you to dip into. So, okay, so we've had a little bit of a whistle-stop tour of perhaps cell biology, and perhaps that's given you a little bit of a thought. But what I'd like to do for the last few moments then is just look at supporting your plans for the next academic year, and really to see what your options are. Well, as you know, at the Trafford College Group, and whether you're studying that at the Stockport campus or the Stretford Road campus, there are 3 main courses. And if I was to describe them starting on the right-hand side, you'll see that we've got our level 1 course, which is applied science. And you can see what the entry criteria are for there. So the entry criteria that we normally look at in science is how you perform in GCSE English language, maths, and science. So we'd look to see how you got on. And if you met the entry criteria for level 1, fantastic, that's brilliant. If, of course, that your grades are a bit higher and you're starting to generally get grade 3s in your GCSEs, then you'd look for the blue course. And the blue course is a 1-year program, and it's the BTEC level 2 in applied science. So you'd be able to go on to that. And if you came out with generally 4s in your GCSEs, well, then you'd be able to go into the first year of your BTEC level 3. Now, in fairness, just to be clear, the BTEC level 3 is a 2-year program. So you need to be making sure that you did well in your first year and then you progressed on to the second year of the course. So they're the 3 main courses that you'd be looking at: level 1, level 2, and level 3. And just remember that level 3's got a second year to it. After level 3, most of our students then go off to university, and they'll be studying subjects such as biomedical science, biology, chemistry, pharmacy, physiotherapy, paramedic studies, quite a wide subject to name but few. So we're now coming into the summer. If during that time there's any questions that you feel that you want to need some support with to help you make a decision, please do get in contact. My contact details are there. I'd be more than happy to help you. If you want just updates, general updates from the college, I've listed our Twitter sites, but if you'd like to email me or call me directly, that's cool as well. Hope you have a really nice summer break. Take care of yourselves, and I'll see you soon. Bye-bye.